Good afternoon, everyone. I'm Kirstyn Bonetti, a member of the Exposomics and Children's Environmental Health Board at Mount Sinai. I want to thank you for joining us for another thought-provoking session of Lunchtime Chats, where we have the opportunity to pick the brains of the world's leading exposomics and environmental health scientists. As we learned in previous sessions, exposomics is all things environmental that we are exposed to throughout our lives: food, chemicals, medicines, and even stress. So today, we'll talk about our gut and the microbiome. We eat every day, many times a day. So what we feed our bodies is key to our overall health. What I think we all want to know is what foods we should avoid and what foods we should eat to better protect ourselves. What is the gut microbiome? Dr. Ryan Walker will explain that today. He is a leading environmental health scientist looking at this, and his unique career path provides him with an unparalleled perspective on how our diet impacts health. He is trained in clinical nutrition, exercise physiology, and the genetics of metabolic disorders. But that's not all. He has worked as a professional chef in some great restaurants, and he knows a few things about getting fit. And now I turn it to Dr. Walker, who is an assistant professor of environmental medicine and public health and a member of the Institute for Exposomics Research. The presentation today will be about 25 minutes, followed by 20 minutes for audience questions. Please submit your questions using the Q&A function. And if we can't get to them all, we will work to get answers to you on social media. Dr. Walker, the stage is yours.
Thank you, Kirstyn. That was a wonderful introduction, and I'm very excited to talk about an often very confusing yet thought-provoking topic, which is the gut microbiome, paired with a second very controversial and often misunderstood topic, which is our diet and how those two things interact with one another to impact our health. So as I just mentioned, there's lots of questions surrounding the microbiome. What is it? What does it do? So I thought I'd lay out a talk today that outlines some of the most common questions related to our gut bacteria and our diets. What is the gut microbiome? How do we study it? How does the gut microbiome develop? And what does it actually do in our bodies? How is our gut bacteria linked to health outcomes? And what role does our diet and what we consume on a daily basis have on the bacteria that reside in our guts? And lastly, some ideas about how we can promote gut health. So what is the microbiome and how do we study it? Well, essentially, it's the entire ecological community of friendly bacteria, commensals, and pathogens that live within spaces on our body. There's 10 times more microbial cells than human cells, so we're vastly outnumbered, and we're essentially hosts providing a space for these communities to live in. So the microbiota are all of the bacteria within a given community on our body site. And the term that you usually hear, which is the microbiome, actually refers to the collective genome, or all the genetic material of bacteria in a given community. So those are two kind of different nuanced words. To put it in context, the human genome has about 23,000 genes in it, whereas the microbiome, for example, in our guts represents almost 3 million genes. So what that essentially means is that there's an unbelievable wealth of different function going on within our guts. Our microbiota produce thousands of metabolites that can influence our fitness, our phenotypes, which is the traits that people can see on our exteriors, like obesity and other health matters like that. So to put it quite bluntly, it's a very complicated system and it's very hard to study. And to put that in context, here's an evolutionary tree representing the domains of life. Here we are, here's animals, and here's plants. Bacteria has an entire domain in and of itself. And when we look at how to study these, they're classified into different levels of taxonomy. from the phylum level. And some examples of that are Bacteroides and Firmicutes, which you've probably heard quite a bit about in media publications and scientific journals, all the way down to the genus and the species level. So what that means is that on each site of our body, there's a completely different microbiome, these communities that populate different sites of our bodies. So the hair, the skin, the vaginal microbiome and the colonic microbiome are all very different from one another and impact our bodies in different ways. Far and away, the most diverse and complicated microbiome is that which is in our colon, our gut microbiome. And the way we study that is we can collect stool samples, and those kind of serve as a proxy when we analyze them for what's going on inside of our guts. Now, the way that we study these samples is a given sample, the DNA is extracted from all the bacteria, and there's two ways to do this. There's the 16S approach, which is a very short segment of bacterial DNA that's unique to bacteria, but there's enough variability within each of those fragments to determine what bacteria is different from another. Those are amplified, processed against known databases, and then we get a representation of the abundance of what bacteria are present. The other method is called shotgun metagenomic sequencing, and that's where we're not just looking at one fragment, we're looking at the entire genome of bacteria. Software is used to process those samples and compare them against reference genomes. And this method, along with giving us abundances and distributions of what bacteria are in a given community, we can also determine the function of what those bacteria are doing because we have their whole genes. So when we have all this information, we understand that individuals differ in the composition often quite greatly of their gut microbiomes. And when we have that information, we can compare unique individual microbiomes with others in a known population to identify quote unquote signatures that may or may not associate with diseases, different traits, behaviors, or any question that you want to study that might be related to the microbiome. So how does our gut microbiome develop and what is it actually doing? Leading research suggests that the influence on our gut microbiome may even start in utero. Maternal diet and environmental exposure and maternal health is thought to potentially influence the very first bacteria that are present in an infant's gut. Then, as children are born, birth type, whether it's cesarean or vaginal delivery, administration of early antibiotics, and there on out, environmental exposures such as family number of people living in home, the presence of a dog, the timing of the introduction of solid foods, and whether or not children are breastfed, for example, influences the development of the early life gut profoundly. And as you can see, this chart illustrates, as we get older, we have a more of an abundance of bacteria, which means generally more. And then we also have a greater diversity, meaning many different subtypes of bacteria are present as we age and have increasingly more environmental exposures to influence them. Now, this is important because a quote unquote normal bacteria tends to lead to normal development. However, when the early life acquisition of microbiota is perturbed or altered, for example, through the administration of multiple low-dose antibiotics for ear infections, or as I mentioned earlier, through birth type, you can have a missing or a dysbiotic bacteria where very critical keystone species may not be present. And that influences the microbiome as we age. And that could lead to altered development of our immune system, which is associated with higher risk for allergies. Changes in our metabolism as we age, which is associated with obesity and cardiometabolic disease, and also changes in our nervous system development, which have been linked to autism. So we actually did some work to try to understand in more detail the earliest factors of the environment that are impacting infant microbiome development. We recruited 40 family trios. We measured the microbiome of the father, the mother, and then the infant when it was born. we sequenced those samples and compared the gut microbiota of moms and the vaginal microbiome and the very earliest microbiome from the infant to try to understand what is contributing to the development of the fetal microbiome and the first presence of bacteria in the gut. Then we measured factors that are known to influence the early microbiome, like breastfeeding status, timing of first solid food introduction, and antibiotics exposure to understand how microbiome develops into early life when children progress from infancy into the toddler stage. And what this shows, this is called a beta diversity plot, which is essentially showing how different each sample in a given person is from all of the other samples in a study that you're comparing. So in this chart, you can see that these colors here represent the fathers and the mothers, and these represent all the infants. So they're separated by space, indicating that they're very different from one another. But when we look at this and introduce time as a component, we can see that the children are becoming more adult-like as they age out to a year. So they start very separate and very different with a low diversity and completely different representation in their guts from adults. And as they progress through life into about 1 year of age, they become more adult-like in the way that their overall microbiome structure looks. This is called an abundance plot, and what it shows is the different ratios of all the bacteria in the gut at the phylum level, which is a more broad way to group the bacteria. And we were able to see that actinobacteria increases, specifically bifidobacterium, increases from day 7 by almost 70% at 4 months. And we think this is directly attributed to breastfeeding because bifidobacterium are one of the most commonly found bacteria in breast milk. As I alluded to before, breast milk is an essential source of microbiome transfer from mother to infants in early life. And it's full of incredibly critical and beneficial microbes from the proteobacteria, firmicutes, and actobacteria families. The variability in what is in mother's breast milk is very high, but you can see they're primarily dominated by proteobacteria and bifidobacterium. And these are common microbes that you hear about in the form of supplements and probiotics, which I'll talk about in a little bit. So to put it more concisely, very early life is a critical time for the development of our gut microbiomes. But what does it actually do inside of us? So the primary responsibility of microbes in our guts is to digest and harvest energy from foods that we can't digest ourselves, like non-digestible starches and fibers. So resistant starch, cellulose, pectins, and unabsorbed sugars are all sources of fuel for the bacteria that colonize our guts. And the byproduct of that is the synthesis of vitamins and critically generation of these metabolites called short-chain fatty acids that are critical not only to the survival of the bacteria ourselves, but they play a critically important role in our own physiological systems. The microbiota also influence our immune systems and protect us against infections by producing antimicrobials themselves that sort of tamp down the colonization of pathogenic bacteria, for example, like E. coli. And they're also linked to the gut-brain axis, which is this communication system that's been identified where bacteria in our guts, by virtue of creating these metabolites and short-chain fatty acids, integrate neural, hormonal, and immune signaling between the gut and the brain. And those are critical in modulating things like our appetite, our response to food in terms of glucose control and metabolizing fat, and they can even influence our behaviors. So taking all the functions of the bacteria into consideration, how is the microbiome actually linked to our health? So one of the primary influencers of the microbiome and the purpose of giving this talk is our diet. So diet is intimately linked with our intestinal microbiota. So as I mentioned before, non-digested food components pass to our colons and influence the diversity and the activity of our microbiome. Those bacteria then transform these food components and produce really critical metabolites like short-chain fatty acids that directly influence not only our GI function, but our health in terms of disease risk. So there's an intimately linked triad between diet, our microbiome, and the health of the host. So altered microbiome populations, they're being increasingly associated with a whole host of diseases like obesity and type 2 diabetes, cardiovascular disease, all the way down to allergy, autism, and cognitive decline. One of the very first studies that identified this relationship was a study in lean and obese twins. And what they were able to show, this is a graph showing essentially the diversity of the gut microbiome in lean and obese twin pairs who are otherwise genetically identical. And you can see that the obese twins had a much lower diversity than the lean twins. And that is a metric that has been associated with poorer health outcomes. Critically, and very interestingly, when they did fecal microbiome transplants into mice from the lean donors and the obese donors, the transfer of microbes into the mice alone was enough to recreate the obese phenotype in the absence of any other bacteria in the mice. So this is showing the microbiome from an obese donor by itself is enough to create obesity in a mouse model. And that speaks to the causal mechanisms of the microbiome in health. So we have all of these dietary inputs that elicit a functional response in our bodies, and those are associated with health outcomes. So I wanted to list a few of those here. So the top level of this image shows the different things that we can consume. Some good ones are fiber, for example, and some bad ones are sugar, saturated fatty acid. And then there's an associated functional response. So bad things that you eat result in lower short chain fatty acid production as opposed to high short chain fatty acid production. And that has health consequences, and some of those that I just mentioned. So our diet is intimately linked to our health through a mediating relationship of the microbiome and the things that they produce in our guts. Some of the critical short chain fatty acids that I wanted to mention are called acetate, butyrate, and propionate. And these things are absolutely essential, not only for the health of the bacterial community, but also for our own physiological health. So what does it mean if you don't have the right bacteria in your microbiota? So there's a homeostasis state where, for example, if you're healthy, you're eating a healthy, varied diet, things tend to stay in balance and you have a higher representation of bacteria that are good for you, mutualistic bacteria like bifidobacteria, lactobacillus, firmicutes, and bacteroides in distributions that are known to be associated with health. Dysbiosis is essentially an increase in pathogenic representation of bacteria, a reduced overall diversity, and specifically the loss of these beneficial microbes that I mentioned here. And what that does is gives a more of an opportunity for pathogenic bacteria to populate and thrive, and that can cause problems. So our daily exposures can influence the balance and overall health of our microbiome in our gut. But what role does our diet have on the microbiome? As I've already mentioned, diet rapidly alters the microbiome community structure. Not only that, but it also can rapidly alter their function in as quickly as 4 days. Our healthy microbiomes can be very resilient to short-term changes, and they're able to revert back to their original state. So the long-term stability to dietary change, that hasn't been studied too extensively yet. But this is very challenging to study. Typical studies look at the association of one nutrient for example, with one bacteria and its abundance and function. The studies that are needed are more complex because our diets are complex, as are our microbiomes. And we are starting to use new adaptive methodologies to try to answer some of these questions more definitively. But as I alluded to before, it's a very complex paradigm. One of the earliest studies that recognized that diet profoundly influences the microbiome compared children in rural Africa who consumed lots of complex carbohydrates and a high amount of plant fibers to children from European populations that are essentially eating a Western diet that was high in fat, high in sugar, and low in fiber. And you can see here that the children in Africa and rural populations had a very high representation from Bacteroides, whereas the children in the European population had a very high representation from bacteria from the Firmicutes phylum. They've created a ratio, the Firmicutes to Bacteroides ratio, which you may have read about, that associates very strongly with health outcomes. This was later confirmed in a larger study. This is just showing the overall abundance of Firmicutes in black and Bacteroides in white. And you can see that the non-Western populations have a much lower level of Firmicutes, which is associated with health outcomes, suggesting that a Western diet, which is known to be not beneficial for our health, it also has profound impacts on our gut microbiota. So as you can imagine, our diet quality optimizes our microbiomes. So when we consume things like fruits, vegetables, lots of high fiber and plant-derived proteins, we have a higher microbial diversity, better immune homeostasis, and essentially a healthy gut barrier and higher short-chain fatty acid levels. Whereas if we have a poor quality diet dominated by saturated fats, refined grains, sugars, and alcohol, we have a reduced microbial diversity, higher levels of inflammation, and low short-chain fatty acids. And that can impact our health. So now we know that microbiome is influenced by diet. A rural diet is more beneficial to diversity, whereas an urban diet high in these dietary components that I just mentioned lowers diversity and increases the amount of non-beneficial species. But emerging work shows profound effects of food additives on the microbiome and also potentially health. And similarly, non-absorbable sugars like sweeteners, stabilizers, preservatives, emulsifiers, food dyes, salt alternatives, pesticides, and organics and pollutants remain relatively understudied. So we aren't really quite sure what role these other components in our food are playing on our gut. One food component that I've spent a lot of time researching is fructose. What this graph shows is the amount of high fructose corn syrup used in the food market as opposed to sucrose, which is common table sugar. And as you can see, there was a huge surge in its production and use from the 70s out to 2006, where it represented more than half the market. Coincidentally, and this is just an association, the boon in the use of high fructose corn syrup also tightly correlates to increases in obesity in our country. And this graph showing the per capita use of high fructose corn syrup per year shows that the US is leading the way. We're consuming lots of this and it's pervasive across our food system. This is important because although fructose is a normal sugar that's naturally occurring in fruits and honey, when it's used in high fructose corn syrup as an additive, the ratio of fructose to glucose is different than that of sucrose, which is table sugar. So there's more fructose used in high fructose corn syrup. This becomes important when you put it in context of consumption and realize that 20% of young adults are consuming 3 or more cans of soda per day. We did a metabolomics analysis of popular sugar-sweetened beverages a few years back and found out that drinks like Mountain Dew, Pepsi, and Coca-Cola have almost 45 grams of free fructose in them. And so when you're consuming 3 drinks per day with 42 grams of sugar in each drink, that equates to 516 calories, which is over 25% of what you should be consuming on a daily basis. So that's representing a massive fraction of our daily consumption. So we wanted to know if fructose was actually altering the microbiome. So we did a short clinical trial where subjects consumed sugar-sweetened beverages for 7 days, and we measured we collected stool samples, measured their microbiome, and tracked their weight. 25% of their diet was represented by added fructose. And what that looks like is this. So each of these rows represents a day. Each of these packets is about 50 grams of sugar, which is what's in a soda. So this is how much sugar you'd be consuming if you drank 3 sodas a day for 7 days, which 20% of the adolescents in the country do. And what we found is that high fructose diet, it does alter the microbiome. You can see that this shows the mean percent changes in the taxa or the bacteria from baseline when they went on the intervention. There was a decrease in Bacteroides with an increase in Firmicutes. That increases the fructose to Bacteroides ratio, which I mentioned earlier, which is a general indicator of microbial health. There was a decrease in Prevotella, which is associated with liver disease. And there were decreases in Prevotella copri and Ruminococcus bromi, which are both involved in fiber degradation and complex starch breakdown. So this is suggesting when you consume this much extra fructose in the form of a beverage, you're displacing the consumption of fiber-containing fruits and vegetables. And that's actually changing the function of your gut. We wanted to know if this was actually causally related, so we did a fecal microbial transplant into mice. This is another beta diversity plot, which is showing the difference of each community structure from all of the others that are measured. The red circle is all of the mice that were transplanted with samples from the participants that were on the fructose intervention. The green cluster is mice that were transplanted with samples from human donors that had not yet begun the fructose intervention. And the white circle is a control group. And you can see that they're separating differently in space, indicating that fructose had an effect on overall community structure of the microbiome in these mice. Interestingly, when we look at the physiological aspects of these mice, we see that those that were transplanted with the fructose samples had higher percent fat, higher liver enzymes, which is suggestive of fatty liver disease, and higher interleukin-6, which is a marker of overall inflammation. So this is showing that just by virtue of transplanting microbes from humans that consumed fructose into mice that don't have a microbiome, called germ-free mice, that transplant is enough to elicit these phenotypic or physiological results that are suggestive of risk for cardiometabolic disease. So now that we've seen that diet can impact the microbiome profoundly, what can we do to promote our gut health? So we can take prebiotics. These are in foods or nutritional supplements that they cannot be digested by humans. So just like complex starches, they pass to the microbes in our gut and give them food to do beneficial things for us. We can take probiotics, which are living microorganisms like yogurt and other supplements. Most of these contain bifidobacterium and lactobacillus species, which have been increasingly associated with health outcomes. Or we can take synbiotics, which are blends of pre and probiotics. There's conflicting evidence of how beneficial these are in actually preventing disease, but the general consensus is probiotics, you can't do any harm by taking them. But what has been shown is that probiotics can improve existing health outcomes like irritable bowel disease, colitis, GI symptoms, and E. coli infections. Most importantly, you should be eating a high fiber diet, rich in vegetables, fruits, and complex carbohydrates. And those also happen to contain polyphenols, which are also beneficial to us and our microbiota. So essentially, high fiber diets selectively are used by beneficial microbes, and that confers a health benefit. And as I've mentioned already, when we're consuming high fiber in our diet, it increases the diversity of our microbiome, which is also associated with beneficial health outcomes. So in addition to eating high fiber, we should be limiting high saturated fat, absolutely limiting added sugar, in particular high fructose corn syrup because of the fructose component. Artificial sweeteners should be avoided, and we should limit ultra-processed foods containing food additives because not enough work has been done to understand how they might impact the microbiome. Critically, microbiota, they should be considered a key aspect of nutrition. We're not quite there yet, and the medical community should adapt education for physicians and counselors to change their public health message to include better representation of the microbiota in our diet and how those things interact to impact our health. So just to wrap things up here, what we know, the microbes in our gut, they influence our energy metabolism, and that can be impacted by diet and medications in a very robust way. Microbiome composition influences response to drugs, and dietary fiber is in particular incredibly beneficial and influences the microbiota composition and has been associated with better health outcomes. What we don't know is if natural probiotics in food is better than actual probiotic supplements, and should we take these preventatively rather than to treat existing conditions? We don't know if microbes are directly influencing our food choices and our appetite through interaction with the gut-brain axis. And we also don't know if low-dose antibiotics in food, not from drugs, but in food, is affecting human health by virtue of changing the microbiome. And as I said before, the effect of pesticides, food additives, exposure to chemicals, medicals, and other organic pollutants in our environment are assumed to impact the microbiome, but the mechanisms in which they do has not been fully resolved. So the future of this relationship between diet and food is essentially in personalized nutrition for health. And to picture what that looks like, you have these people that eat different ratios of fat, protein, carbs, and supplements, et cetera. You can measure the microbial response, and then there's an associated host response. But then you can use these microbiota responses that are very individual to predict how people are going to respond to the diets they consume. And from that, you can create personalized diet designs that are individualized, not only to each person's physiology, but also to their microbiota. And that's sort of the future of combining these two domains. There was a very recent article, which Carla was so kind to share with me, in the New York Times called "How Healthy is Your Gut Microbiome?" I put the link up here because it's actually a very nice primer that answers a lot of the common questions that I may have went over too quickly today. So this is a great resource to answer any questions that you might have that I'm not able to answer at the end of the talk. And I'd be remiss if I didn't acknowledge the team that's helped with all of these research avenues that I pursued. It's a team science, and I couldn't do it without them. I wanted to thank the organizers for the opportunity, and now I'm happy to take any questions.
Thank you, Dr. Walker, so much for a truly excellent presentation. Before we start the Q&A, I invite you to visit our website, MountSinaiExposomics.org, to find out more information and resources about how the environment impacts our health. And be sure to follow our social media accounts at Sinai Exposomics to get real-time updates. If you have any more questions or concerns in your community, from questions about pesticide use to healthy foods to clean water, don't hesitate to contact us. As a member of the board, I invite you to get more involved. We are seeking nominations for the Mount Sinai Exposomics and Children's Environmental Health Board, a new board that will drive philanthropy and the work of the Institute. Please contact us to nominate someone and to learn more. So now let's turn to the question and answer section. So first, can you go back to the beginning of your presentation? You talked about exposure to dogs and the impact of having pets. How does that impact the microbiome?
So it's a great question. having a dog or a pet of any kind in the home when you have an infant in early life increases general exposure to more diverse sources of the microbiome. So as any parent knows, children can't keep their hands off anything. So everything they touch gets up to their mouth, and that includes touching the family pets, getting licked by the pets, touching things on the ground. And those are all sources of exposure to environmental bacteria, which help to populate and colonize and increase the diversity of the gut microbiome in infants. And increased diversity is associated with better health outcomes in the future.
Okay, so it sounds like we should all go out and get pets.
Yeah.
The next question, is there a strong correlation between oral and gut microbiota? And if so, could oral samples be used in human studies?
So oral samples are studied extensively in human studies. They have very different function than the gut microbiome. So the bulk of literature looking at oral samples relates to oral health, because as I mentioned, each microbial community it developed specifically in that specific site in which it populates. So the function and the structure of the microbiome in the mouth is going to be profoundly different from that of the gut. So it depends on the scientific question, but generally oral microbiome are used to study oral health, like risk for dental caries and questions like that.
Wonderful. Okay, so on to the next question. Can you talk about sugar substitutes and sweeteners, things like xylitol or agave?
Yeah, so sugar substitutes, in particular, non-absorbable artificial sweeteners that are used to sweeten foods, are a big problem in human health. There's been a few pretty convincing studies that have showed recently that consumption of artificial sweeteners increases risk for type 2 diabetes and cardiovascular disease. And some other recent work suggests that may be by virtue of the fact that they're non-absorbable. So when you consume a non-absorbable sweetener, you can't absorb it like you do a regular sugar, like for example, glucose. So you can imagine what happens. It gets down into your colon, and the bacteria that populate your colon, they see that, and then they metabolize it and consume it. And it can result in the generation of non-beneficial metabolites, and it can also change the structure and the function of the gut bacteria just by virtue of consuming it. There's no definitive consensus on the mechanism of that action yet, but in particular, non-absorbable sweeteners, I would recommend avoiding them. The fructose work that I've been doing is essentially representing the same mechanism. We can't absorb that much fructose when we consume it, so it's non-absorbed. It goes into our guts and can change our microbiome function, and thereby influencing our health.
Great, thank you. And do you recommend an organic diet?
Organic diets are ideal, yes, but cost and expense can be very prohibitive, and even I experienced that myself. So an approach that I would suggest is picking and choosing specific foods that are known to be sprayed with more pesticides or chemically treated, like berries and leafy greens and other apples and other produce, and focusing on those to reduce costs. So that it's more likely that you're able to sustain those changes in your diet. Any changes in our diet should really be focusing on things that are manageable and sustainable for the long term. Otherwise, they aren't going to really impact our microbiome in the short term, or our health in the short term. So if you want to go organic, but you can't, pick and choose the things that you're poised to benefit from the most.
Great, thank you so much. The next question is there a test to check your microbiome? So can you look at the good versus bad bacteria, say in a stool sample?
Yeah, there is. There's a lot of mail-in tests that you can do. What you get out of that is a comparison of you against a giant database of other people. So there's a lot of companies that are trying to use the microbiome to actually diagnose and actually report back your risk for certain diseases, just in the same way that 23andMe is trying to do the same thing with the human genome. But you have to be really careful because the information that you're getting back doesn't necessarily mean that you're going to get diabetes or become obese by virtue of your human genome. And the same can be said for the microbiome. There's not enough work done on how our diets are influencing the microbiome. It's a very complex analysis, like I referred to before. So what you get back from these tests is what you look like compared to everyone else, and that can give you sort of a picture of what your gut health may look like. How useful it is right now in determining your health and your risk for disease, that's still up for debate.
Great, thanks so much. The next question is, what about probiotics? Are all probiotics created equal? So unrefrigerated, refrigerated probiotics. Which one should I grab?
So I mentioned this, and maybe I went over it a little quick in the talk, but probiotics have been shown to have very, very efficacious results on treating conditions that you might already have, like gas, bloating, diarrhea, irritable bowel disease, and things like that. They're primarily composed of different mixtures of species from the Lactobacillus and Bifidobacterium families. Those are the two taxa that are most extensively researched. And the general thought is they're not going to do anything bad to you if you take them, and they could confer some benefit. But whether or not taking them prophylactically to mediate future health risk is going to work, we don't know that yet. So the frozen versus fresh, I don't think that plays a huge role in the impact of probiotics. So it depends on what the intended use is. They can be useful, but we don't know enough about them yet.
Great. And is there any research on the influence of the gut microbiome on neurodevelopment?
There is. That's a little bit out of my domain of expertise, but I referred to the gut-brain axis, in which metabolites that gut bacteria produce interact with cells in our guts that can increase or decrease appetite hormones, for example. And so our satiety, our fullness, and our appetites and our cravings for food can be influenced by our microbiomes. There has been studies, many studies linking early life microbial diversity and development to neurodevelopment and cognitive outcomes. But that's not my general area, so I don't have enough expertise to comment on that.
No problem at all. Is there any research on microbiome health and autoimmune disease? I know you mentioned it briefly at the beginning as well.
There is. There's extensive research on microbiome and autoimmune disease. And it's thought that if your microbiome early in life is perturbed by some external influence like antibiotic exposure, cesarean versus vaginal delivery, or unhealthy diet as you develop, your immune system can be impacted as a result. And so an example of that is children that were born cesarean and not vaginally have a different microbiome as they age, and they're more prone to allergy and dermatitis and atopy and diseases like that are linked to autoimmune disorders.
Wonderful, thank you. And then if you're taking antibiotics for an extended period of time, perhaps after surgery or after a long infectious illness, what's the best way to rebuild the microbiome? I think you're probably going to say through diet, a good daily diet, but I'd love to get your thoughts.
Yeah, so I don't want to vilify antibiotics at all. They're critical for human health, but they are extensively overprescribed in the medical healthcare setting. And they do drastically perturb our guts. And I think anyone that's taken a course of antibiotics you can relate to that. And Kirstyn, you're right. The best thing that you can do is, even before you're taking the antibiotics, increase your fiber intake. This is an example of where probiotic supplementation has shown to have some impact. And the other things that I mentioned, like having diverse non-digestible starches in your diet, can all help sort of bring back your keystone populations and your microbiome set point from being leveled by antibiotic doses.
Thank you so much, Dr. Walker. We're at the end of our time now, and we really appreciate your talk today. It was truly, truly very interesting. And we will work on developing a new resources page to answer the many questions that came through today. And we appreciate all the questions and everybody's time today. And just before we close, we'd ask you to complete an evaluation survey. We'll put the link in the chat and email it to those who have registered for this talk. And please note, our next chat is next Wednesday, March 8th, with a talk by Dr. Rosalind Wright on climate, air pollution, and increased lung disease, the increased lung disease risk. So everyone, we thank you again for your time, and please enjoy the rest of your day.